Hi there! Let's understand the Golgi apparatus, also called Golgi complex or Golgi body, is like a busy post office or packaging and sorting factory inside the cell. It receives proteins and lipids that are made in the endoplasmic reticulum, especially the rough endoplasmic reticulum, then modifies, changes or improves them, sorts them, and packages them into little delivery vehicles called vesicles to send them to the right places inside or outside the cell. Think of it this way. Rough endoplasmic reticulum makes raw proteins, like uncooked food. Golgi, kitchen plus packaging department, cooks, modifies, labels, and packs them into boxes, vesicles, for delivery. Here are the key parts and how they work. Structure. It looks like a stack of flattened pancakes or pouches, called cisternae. Usually 4 to 8 cisternae stacked together. This stack has two different sides, polarity, very important. Cis face, entry side or forming face, convex, curved outward. Closer to the endoplasmic reticulum and nucleus. This is where incoming vesicles arrive from the endoplasmic reticulum. Trans face, exit side or maturing face, concave, curved inward. This is where finished products leave in new vesicles. In between are medial cisternae. Surrounding the stack are lots of small and large vesicles, tiny bubbles made of membrane. There are also special regions. Cis Golgi network, area before the cis face. Acts like a reception desk that checks and selects what should go forward and sends wrong things back to the endoplasmic reticulum. Trans Golgi network, area after the trans face, acts like a sorting and shipping department that decides where each package goes, for example, to lysosomes, plasma membrane, or outside the cell for secretion. Here is an illustration to help visualize it. How things move through the Golgi. Proteins lipids are made in rough endoplasmic reticulum, wrapped in COP2-coated vesicles, travel forward, anterograde transport, fuse with the cis face. Inside the stack, the material moves from cis, medial, trans cisternae. This can happen by small vesicles butting off one cisterna and fusing with the next, or sometimes the cisternae themselves mature step by step. At each level, especially medial and trans, special enzymes in the Golgi add or remove sugars, glycosylation, like decorating the protein, add phosphate, phosphorylation, add sulfate, etc. These changes are like adding address tags, or making the protein work properly. At the trans face or trans Golgi network, the finished products are sorted. Some go into secretory vesicles, fuse with cell membrane, release outside, secretion, for example, hormones, enzymes. Some go to lysosomes, for digestion inside cell. Some become part of the plasma membrane, or other organelles. Sorting uses signal sequences, like postal codes, on the proteins. Many outgoing vesicles get a clathrin coat to help them form and travel along tracks, microtubules, to their destination. Other important points. Traffic is two-way, forward, endoplasmic reticulum, Golgi, and backward, Golgi endoplasmic reticulum, using different vesicle coats, COP2 for forward, COP1 for backward. This allows recycling and quality control. Golgins, long protein fibers that hold the stack together like a scaffold and help guide vesicles. The Golgi is very active in secretory cells, for example, cells that make saliva, hormones, or digestive enzymes. In short, the Golgi apparatus is the cell's modification, sorting, and dispatch center. Without it, proteins would be unfinished and go to the wrong places, like mail without addresses or wrong packaging. This is why it's especially prominent, big and easy to see, in cells that secrete a lot of stuff. I hope you liked the video. Please subscribe this channel and press the bell icon.